I don't like the position of this. Very annoying. Okay, better. Oh boy. Yes, everybody. Can you guys see me? Yes? I don't know what my camera is doing. Oh, okay. That's good. Good for you. We can see. Okay. How many are we? 20. Where is everybody? The people that did the psychology exam, how was it? Good? A quiz? Thought it was an exam. It was good. Okay. Where's everybody? How many are we here? 21. 'Say good night. 25. 10:02. I'm trying to give them like 5 minutes. Twenty-six. Hello. How are you doing? Ten oh four. Okay. 1 minute more. 28. $29. 10:05. Okay. So this will be a very fast review. You understand? The reason is because I already done this review, all right? So you can always go to the YouTube lives and look at it. And most importantly, there are videos for you, all right? So you have more than enough resources. So this review will be very brief, and we do a second one on Sunday, probably. Just two is enough. For the last two chapters, that's special senses and autonomic nervous system. I'm not doing review on them. We are sleepy. Just did lecture. If you are sleepy, put your leg inside water, all right? Or you can go sleep. Maybe later you can just watch it. It's not a big deal. For the last two chapters, autonomic nervous system and special senses, I made videos for them. You use those videos, please. Go and watch it. There are questions there. And those questions are seriously important. You understand? Because, yes, of course, put your leg in the water. Those days, this is when we come back from posting. You understand? You have to read between now and like 1 o'clock. Then you sleep and wake up by 6 o'clock. It's crazy that year. So, but what I'm saying is that we don't need to waste our time doing review on those two chapters, because I've already discussed the same questions on those videos. So I want you to watch those videos. I'm supposed to post the autonomic nervous system video today. I'll post it just after discussing with you guys. And sorry, I have not posted it since because my laptop is having a problem. In short, I have to get laptop from someone because I purchased a new one, and Apple, they are taking 1,000 years to send it to me. I don't understand. Okay, so let's start with we are still 28. We are supposed to be up to 50 or more in this review. Because people that sent me message, we are almost 60 people. So I don't know how you guys become 28 all of a sudden. Okay. Which topics do you think you have more need for? There are a lot of topics. Which one do you think you have more need for? Once we should start with chapter 1 to 3. Good. I have that in mind as well. Yes. It's so long ago, and I didn't do any distinct video for chapter 1. See, I want to tell you that all the chapters we have questions from them. I understand. I have wide range of options. For people that are in my class, I have wide range of options to ask questions. So bone and muscle. Bone is number 7. Muscle is it? Which one did we do? Muscular system or muscular tissue? Let me see. Tissue, okay. So we did one special senses is two out. Okay, so we can easily do like 5 chapters today. Very fast. Then we do the rest tomorrow on Sunday. Okay. So let's start with chapter 1. Let's just go downwards like that, all right? And stop anywhere we are tired. I want this review to be roughly 2 hours or maybe 3 hours, but 2 hours is okay. We can't kill ourselves. 27. Are we going backwards or forward? I'm saying all these things to wait for these people, but okay. So I think you should have avoid chapter 1. All right. Chapter 1 is filled with history. I think you should avoid chapter 1. Somebody's happy. Why are you being happy there? They say why? Because I said so. They say why? Or you want to do chapter 1, eh? It's laughing. Yes, now. Okay, so we are starting with chapter 2. Yeah, too much. I don't like history. Like, I really don't like history. I'm even trying to remember my date of birth. Date of birth. Birth. I don't know the pronunciation. I'm trying. Then I'll now start. Exactly. Exactly. Say we don't need we. Just say I. Say my birthday. Okay, guys. So let's start. We're starting with macromolecules, all right? That's chemistry. So please, everybody has to attempt. We are very few here. So I sometimes it's actually better being few because it means that I will know how many people that respond. August 25. Did I tell anybody my date of birth? Hey, we have detectives here. Please, let's start. Please, let's start. All right. Okay, so sodium. Sodium has an atomic number of 11 and atomic mass of 23. Sodium has how many dash? I'm not going to pose the options, all right? So you listen carefully. Sodium has 12 neutrons and 11 protons. Sodium has 12 protons and 11 neutrons. Sodium has 12 electrons and 11 neutrons. Sodium has 12 protons and 11 electrons. Sodium has 12 electrons and 11 protons. Answer quickly, guys. Quickly, guys. Energy. Energy, guys. A is 12 neutron, 11 protons, 12 protons and 11 neutrons, 12 electrons and 11 neutrons, 12 protons and 11 electrons, then 12 electrons and 11 protons. People has forgotten. A lot of people has forgotten this thing. Okay. There are a few people attempting. Listen, this is the rules, guys. I know we are starting, so the energy is not high. But this is the rules. When I ask question, I expect everybody to attempt it. If you don't know it's right, not sure. Don't just keep quiet. If I have 28 people here and I'm getting only 7 responses, it doesn't help me. All right. All right. So please, and the faster we move, the better for us. If everybody answers and I go to the next question, we'll be able to, move faster. Do you understand? I just want to discuss maybe 20 questions from each chapters for you. All right. Just 20 questions, not roughly give or take each chapters for you. So you need to pay attention and you need to work with me. Sodium has an atomic number of 11 and an atomic mass of 23. It means that sodium has 12 neutrons and 11 protons. All right. Atomic number of 11. Atomic number is number of proton. Number of proton is equals to number of electron. So if it has atomic number of 11, that means it has electron of what? 11. You understand? Then atomic mass is the number of what? Proton and what? Neutron. Proton and what? Neutron. So since they gave you mass of 23, that means if you minus 23 from 11 from 23, you will get what? 12 neutron. That's why we have 12 neutron and 11 proton. All right? Okay. Number 2. The chemical properties of an atom are determined by its dash. A, proton. B, electron. C, neutron. D, protons and neutrons. E, particles. Quickly, guys. Quickly. Testing the microphone. Quickly, guys. Exactly, electrons. All right. The answer to that question is electrons. This is a rapid review, so I'm moving very fast. All right. If I just get at least 12 responses, we are 34 here, so 15 responses, at least half of the people respond, I move. All right. So look at this question quickly. Sodium atomic number 11 reacts with chlorine atomic number 17 to become stable. In the reaction, sodium will dash, while chlorine will dash. A, accept 1 electron, give up 1 electron. B, give up 1 electron, accept one proton. C, share 1 electron with chlorine, share 1 electron with sodium. D, become an anion, become a cation. E, give up 1 electron, accept 1 electron. Quickly, guys, quickly. Let's move. Perfect. You guys are so correct. The answer is E. Give up 1 electron, accept 1 electron. Good. I love this. This is what I need. Energy is coming back. You see how fast I am now. So oxygen has an atomic number of 8 and an atomic mass of 16. How many valence electrons does it have? Quickly, guys. Answer. How many valence electrons does oxygen have? How many, guys? Options 2, 4, 6, 8, 16. Oxygen has an atomic number of 8 and an atomic mass of 16. How many valence electrons does oxygen have? Quickly, guys. Perfect. You guys are so correct. You guys are so correct. Yes, the answer is 6. You guys are so correct. Look at this question. What type of bond attracts one water molecule to another? You should attempt this. This is final exam prep. You should attempt these questions. You understand? Attempt it by yourself. If you fail it, no wahala. All right. Which type of bond attracts one water molecule to another? Perfect. Hydrogen bond. This is what I need. When jumping into water, you notice resistance. The resistance is caused by water's dash. Is it adhesiveness? Is it cohesiveness? Is it hydrophobic tension? Is it hydrophilic tension? Is it osmotic equilibrium? When jumping into water, you notice resistance. The resistance is caused by water's what? Is it adhesiveness? Is it cohesiveness? Is it hydrophobic tension? Is it hydrophilic tension? Is it osmotic equilibrium? Quickly, guys, quickly. Show me what you've got. Now it's not time to check slides for answers. Now it's time to just answer straight up. This is last minute prep, all right? I'm going to give you roughly 20 to 25 questions in each topic. You guys are so correct. So the answer is what? Cohesiveness. All right. Cohesiveness is that force between water molecules. All right. We have cohesion in my family. All right. But when I want to hold to somebody else's family, it becomes what? Adhesion. All right. So when water is holding this wall, when you pour water on the wall, you get it's what adheres to the wall. All right. But within each other is cohesiveness. So you have to break their bond. You understand? Before you are able to do whatever you want to do. Okay. So look at this question. Hold on. So look at this question. Sorry, I'll be lagging a little. Anytime I share something like this, I'll lag. So these are the options. These are the options. So blood contains sodium chloride, protein, and cells. The sodium chloride is in A or and dash. The protein is in A or and dash. The cells are in A or and dash. Emulsion, solution, suspension, solvent, emulsion, colloid, colloid, suspension, solution, suspension, colloid, solution, solution, colloid, suspension. Quickly, guys, what's the answer? Yes, the answer is E. E is what? Solution, colloid, suspension. All right. If you don't know how we got it, go and look at the video. If you don't know how we got it, go and look at the video. All right. Look at this question. Look at the options here. Which of these has the highest hydrogen concentration? Look at the options. Lemon juice pH 2.3, red wine pH 3.2, tomato pH 4.7, saliva pH 6.6, household ammonia pH 10.8. Answer, guys. I like the way you guys are answering this thing. Energy, energy, energy. Good. So the answer is A. You guys are so correct. The answer is A. Lemon. All right. Look at this question. That's the options. In a workout, your muscle cells produce lactate, yet you maintain a constant blood pH because A, metabolic acids are neutralized in muscle cells before released into the body. Metabolic bases are produced at the same rate by muscle cells to neutralize the acids. The respiratory system removes excess hydrogen from the blood before pH is lowered. The body maintains chemicals called buffers that resist changes in pH. The last one, endothelial cells secrete excess hydrogen to prevent a decrease in pH. Which one is the answer? Yes, the answer is D. The body contains chemicals called what buffers that resist changes in pH. How does it resist the changes? If there is it's too acidic, it starts mopping up what hydrogen. All right. Even the kidney is also part of the buffer system because it pushes what hydrogen out through the urine. And if there is too much basicity, it does what it mops out what OH. All right. Okay. So let me see. Just a minute. Okay. Yeah, the answer is buffer or buffer. Okay, you guys said it's buffer. So look at this. The breakdown of glucose to yield carbon dioxide, oxygen, and ATP can be described as dash. Anabolic and endogonic, catabolic and hexagonic, anabolic and hexagonic, catabolic and endogonic, anabolic and exothermic. Okay, So you guys are so correct. The answer is catabolic and hexagonic. Very, very, very correct. You guys are doing quite well. Look at this question, guys. Look at this question. Look at the options. Which of the following equations depicts an exchange reaction? Exchange reaction. Yes, you guys are absolutely correct. That C is the one that depicts what? An exchange reaction. All right. Look at this question. Look at this question. Please, I want you to know that all the numbers are subscripts. Subscript is one down, right? All the answers are subscript. Which of the following is not an organic compound? Which one? Which one is not an organic compound? Which one is not an organic compound? Where is everybody? Yes, you guys are so correct. The answer is B. All right. The answer is what? B. So look at this question. A dash. Yes, because it doesn't have carbon. Yes. Yes, I didn't mention that. Yeah, you're correct. So as far as it has carbon, it's an organic compound. Okay, look at this question. A dash reaction breaks a dash down into monomers. Hydrolysis polymer, dehydration synthesis molecule, dehydration synthesis polymer, polymer molecule, condensation reactant. Quickly, guys, quickly. I need you to answer the question as if you're in the exam hall, all right? Nobody's going to assist you, so better not follow anybody and pick the right thing. Two UH-60 Black Hawk gunships hit the target over 8,000 rounds. What's that? Is it my TV? From kills. But during the sweep. Sorry for that. But how is it that my TV will just suddenly start playing? Terrible. Yes, you guys are so correct. Anyone that has hydrolysis, when you pour water on something, it breaks the bond, all right? Hydrolysis breaks the bond, all right, of polymers. Very, very important. Very, very, very important that you know that. So look at this question. I think I have two questions to go for this chapter 1, because I want to do it each of the chapters 30 minutes, at least 34 minutes. But we started by 10:07, so I still have like 3 minutes on it. But it depends on how fast you guys go, because I really want us to go very fast. Look at this question, guys. Look at this question. Look at this question. The formula of an amino acid is dash. The formula of a carboxyl group is dash. Quickly. This one is a giveaway now. Perfect. Perfect. You guys, I like the response. I am feeling you guys. Everybody is really ready for this exam. No dulling. Look at this question, guys. Yes, you're so correct. That the D is the answer. You're so correct. Look at this question, guys. Table salt. Table salt is what? Is it maltose containing glucose and sucrose? Is it sucrose containing glucose and fructose? Is it lactose containing glucose and galactose? Is it glycogen containing glucose and fructose? Is it glucose containing galactose and fructose? Perfect. You guys are so good. Last question. Proteoglycans are composed of what? Proteoglycans are composed of what? You guys are so correct. The answer is C. The answer is C. You guys are so correct. I am feeling everybody. Hey, I love this. Should I bring it on? Should I bring it on? Somebody texted me. The said, "Bring it on, bring it on, Nchebe. Do your worst." I thought you'd be like, "Yes, bring it on. We are too ready for this exam," and you're saying, "No, come on now, come on, come on, guys." Okay, so before we go to the next chapter, in short, what I will be doing now before I go to any chapter is I want to do an advert. So you see this shirt? Somebody gave me this shirt, and it's so nice, honestly. Like, since I was given this shirt, I've not even worn it, honestly, because I was too big. Like, I lost weight. I was dieting, I lost weight. So today I wore it, and it entered me well, and I loved it. Protects me from cold. I was not wearing anything inside, and I was outside. You get? So this is it here. Look at it. Uh-huh. I don't want to turn my back because it's written at the back. So the Instagram handle of the person is bad plugs, like BADPLUGZ. In case of you guys want to patronize her, she's a very good friend of mine, and it's very affordable. Very, very good material, very affordable. Let me write the handle so that's bad plugs. Go there, go to Instagram and check it out. Yes, so this is it. Go to Instagram and check it out. So nice, honest, and it's very affordable. Something you can give to someone. You can give it to me as a gift. Please buy blue. I already have black, so buy blue. Okay, I'll be advertising it every time I jump into the next chapter. I don't care what anybody says. I want to say no pics right now. Is this an advert? Yes, it's an advert. You never get cool. I'm serious. Okay, so I never get. Okay, maybe. But funny thing is that I was wearing it today alone. I went to Queens today. I had a meeting with Professor John Dunny. You will see him. He's a senior. He has he has the most massive lab I've ever seen. I really want to work with him. You know, he taught me around the lab. And for you guys that wants to go into research, there are some professors that you really need to partner with. He's in Queensboro. I went there and I was wearing this, you know. I didn't feel any cold. And I was asking myself, why? Okay. That's by the way. He said, I can't believe this. Please believe it. Believe it. Go to that Instagram handle and go and purchase one from my friend. If you have a business, bring it for me. But you have to give me a sample like this. I'll test it before I tell people. You know, I have markets, right? I have 50,000 people as a market. So I can't believe this. Better believe it though. Okay, so guys, let's move. We discussed the cell chemistry of life. Let's discuss. Sorry. Somebody was asking me about picture. In the next review, we will do some important pictures. Do you understand? There will be pictures on your exams, but you know it's selective. In the next review, remind me, because that next review will be longer than this. This one, I want to time it 30 minutes for each. I don't want to overwhelm you. And I really want you to focus on some of those questions and understand why I presented those questions. But most importantly, you see those videos I was posting in your modules, go and watch it, especially the two last chapters. The two last chapters, I've not posted the last one, the autonomic nervous system. Go and watch it. Watch it like 10 times. Okay, so let me move. Cellular. We did cells, right? The next chapter is cells, correct? Please, you have to guide me because sometimes I'm a little confused about the particular class I'm discussing with. The next one is cells, correct? Please, so one person say yes or no, so that we know what I'm doing. Should be cells now. Yes. I was wondering, why is everybody quiet? Okay. Okay, so let's go. Hold on, I need to take note of what we are doing. So the first thing we did was macromolecule. Just a minute, please. I'm trying to make sure. Okay. Okay, let's go. So if you are very fast, we'll finish cells in 30 minutes and we move to the next one. All right? So let's go. These are the options. What are accumulated cell products that are never enclosed in a unit membrane called? Accumulated cell products that are never enclosed in a unit membrane, what are they called? Organelles, inclusions, microtubules, microfilaments, centrioles. Options: organelles, inclusions, microtubules, microfilaments, centrioles. Give me that energy, guys. Give me that energy. Everybody. No, I'm not showing the screen. I want you to look at me. I know. You can see me, right? Just, I want you to be looking at me. Don't mind me. I show the screen when the question is too long and when I cannot start posting it one. It makes me faster. But when I share the screen, it's always lagging this particular laptop. So you have to bear with me, all right? Until I get the new one. I think by then you guys must have finished the exam. So what are the accumulated cell products that are never enclosed in a unit called? They are called what? Inclusion. Perfect. Perfecto. Perfecto. Look at this question. So now that I'm not posting, you have to pay attention. Which of the following is not considered an inclusion? Okay, you want me to post? Fine, I'll share. But the only problem is that I'll be lagging. That's just what I'm afraid of. I'll be lagging. But if it's... So which of the following is not an inclusion? We have lysosome, we have fat droplets, we have glycogen granule, we have bacteria, we have dust particle. Oh, somebody answered before the... Oh, okay. You have watched that video, right? That's good. That's good. That's what I wanted. I wanted you guys to watch those videos very well before we do the revision so that you'll be pouring me answers recklessly. Come on, guys. Why is the answers few? We're up to 50 here. Oh, yeah. Since we're up to 50, I need 20 answers. Very fast. If you are fast, we can finish up. I'm giving you the most very specific questions. The rule is that everybody must respond. That's the rule. Everybody has to put an answer. Type. Your hand is not paining you. Perfect. Yes, the answer is A. All right. Lysosome is... What did I press? Okay. Lysosome is not an inclusion. Okay, look at this question, guys. What was the answer to this question? Which of the following organelles sorts, cuts, splices, and packages newly formed proteins into membrane-bound vesicles? Come on, guys. Come on. Hit it. Let's go. We have the smooth ER, we have the rough ER, we have the Golgi, we have the Golgi, we have the mitochondrium, we have the nucleolus. Which one? You guys are so correct. The answer is Golgi. I told you the Golgi is the is it Amazon? Yes. Did I call it Amazon? Yes, something like that. Okay. Amazon warehouse. Okay. Somebody said it. So it's Golgi. So look at this question. Perfect. Amazon. Yes. You remember. Okay, look at this question. So yeah, that question. Let me give you the options. These are the options. Which of the following organelle is not involved in protein synthesis? So look at it and tell me which one is not involved in protein synthesis. Quickly, guys. Quickly. The person that put question mark, is there something confusing? Something wants me to explain? Please, if we are discussing, if there's something wants me to explain, don't hold back. Please, you have me now. Enjoy me while you still can. Yeah, dude. Yes, you guys are so correct. The answer to that question is smooth ER. I meant to write that with my answer. Okay, the answer is smooth ER. All right, smooth ER, because smooth ER is concerned with lipids. That's why it's smooth. It's smooth. All right. Aha. I'm actually enjoying this. So look at this question, guys. Which of the following Decomposes fatty acids and detoxifies alcohol, free radicals, and drugs. Which one? Option: lysosome, microvilli, inclusions, perisosomes, Golgi, vesicles. Quickly, guys. Quickly, guys. Okay. Sense. Yes, the answer is perisosomes. The answer is perisosomes. Look at this question. Look at this question. What's the answer? I'm not supposed to give you options for this question, but just for time's sake, like for me to be faster, I'm going to give you option. Just for us to be faster. To reduce the amount of thinking somebody will be thinking right now. What's the answer, guys? If you are participating in answering these questions, you won't be feeling sleepy. Trust me. You'll be gingered. Your sympathetic nervous system will be in action right now, in full flow. A lot of adrenaline will be produced currently, right now. You understand? So, can you attempt the questions? Don't feel, oh, I know it. Oh, I've seen it before. Definitely, you've seen it before. This is just consolidation. I'm not teaching. Nah. Yes, you guys are so correct. The answer to that question is B. The Golgi complex and the rough endoplasmic reticulum. How about this? Which function would immediately cease if the ribosome of a cell were destroyed? Huh? Tell me, guys. You are waiting for option. I see option here. Which function would immediately cease if the ribosome of a cell were destroyed? Quickly, 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 quickly. Exactly, protein synthesis, because protein synthesis happens where? In the ribosomes. Look at this question. Yeah, you guys are so correct. The answer to that question is protein synthesis. Which of the following is not a membranous organelle? Which one is not a membranous organelle? I don't understand why we are 32 here, but it's okay. Which of the following is not a membranous organelle? We have the mitochondria, we have the ribosome, we have the endoplasmic reticulum, we have the Golgi complex. Which one is not? Which one is naked? I said naked, somebody. Jesus, what is this boy saying? Jesus, Jesus. Yes, the answer to that question is D. That's protein synthesis. Come on, guys. Come on. I'm seeing only 5 people. Why? Where is everybody? Where is everybody? Why are we only 29? Yes, you guys are so correct. Ribosome is the naked guy. Eh? He said they tried. Tried what? What did they try? Look at this. What is the answer, guys? Say they tried. A patient was severely dehydrated, losing a large amount of fluid. The patient was given IV fluids of normal saline. Normal saline is dashed to your blood cells, and it's about dashed and sodium chloride. Which one? Yes, the answer is the answer. Let me see. Yes, isotonic 0.9. I told you anything between 0.5 to 0.9 is iso. Anything less than 0.5, if they say 0.4%, then they're talking about hypo. If it is above, maybe they say 3% glucose. You understand? In that fluid, it is hypertonic fluid. All right, that's how you know it. That's how you know it. It's as simple as that. Okay, like this option now that they say hypotonic 0.9. 0.9 can never be hypotonic. Do you understand? Then they say hypotonic 9%. No, 9%, anything above 1% is what? Is hypertonic fluid. So that's how we do the mathematics. I know you guys know these things. So you are very correct. Look at this question, guys. How many minutes do I have for this? 58. I have like 10 minutes for so. Quickly, guys. I really want to keep my 30 minutes per this thing. So that's why I'm doing this. So we can use less than. The number of particles of a solute in a solution is described by the its dash. The ability of a solution to affect the fluid volume in a cell is referred to as its dash. Quickly, guys. Quickly. Move it. Quickly, guys. Yes. Let me see. What are they answering? Somebody said D, somebody said C. Why? The answer is C, guys. The number of particles is osmolarity. All right. Tonicity is, how we refer how that particle works. That's why we say, oh, this fluid is hypertonic fluid. Hypertonic fluid. Oh, that means if I put this fluid, it's going to draw water. This fluid is hypotonic fluid. That means if I put this fluid, it's going to move to the other part inside the cell. Do you understand? So that tonicity is how it affects fluid volume. But the particle volume is what? Is osmolarity. So you guys have to remember this. I have very okay. Look at this question. Yes. Look at this question. Hold on. Okay. So look at the question here. There are two options that you need to choose from. Look at the options here. So a red blood cell is placed in a hypertonic solution. This means the concentration of the solutes in the solution is dash than the concentration of solutes in the intracellular fluid and will cause the cell to dash. Come on, guys. Let's go. Let's go. What's the answer? My people. Come on, come on, come on. We have a few minutes to finish cell. Is that all? Is that all I'm getting? Exactly. So that hypertonic fluid means that there is a lot of what solute in it. So the solute concentration is higher, right, than in intracellular fluid. So what will it do? It will start dragging water out and make the cell to shrink. So the answer is C, higher and shrink. Higher and shrink. Look at this question, guys. Quickly, guys. What's the answer? Quickly. Quickly. Somebody should surprise me. Two solutions are separated by a selectively permeable membrane. If solution A has a higher concentration of a non-permeating solute than solution B, then dash. The solute will pass down its concentration gradient from solution A to solution B. The solute will pass down its concentration gradient from solution B to solution A. Water will pass down its concentration gradient from solution A to B. Water will pass down its concentration gradient from solution B to A. Neither the solute nor the water will diffuse. Quickly, guys. Quickly, quickly, quickly. Quickly, oh, quickly. Where is everybody? Yes, the answer to that question is D. Water will pass down its concentration gradient from solution B to solution A. The answer is D. All right, the answer is D. But this one, I'm not giving you option. Which of the, okay, I will give you option. Which of these processes brings droplets of extracellular fluid into a cell? We have phagocytosis, we have receptor-mediated endocytosis, we have transcytosis, we have pinocytosis, we have exocytosis. What's the answer, guys? Talk to me, people. What's the answer? We have phagocytosis, we have receptor-mediated endocytosis, we have transcytosis, we have pinocytosis, we have exocytosis. Yes, you guys are so correct. You know, cell drinking, you remember cell drinking, pinocytosis? Aha. How you remember cell eating, that's phagocytosis. All right. Remember those things. Both of them are example of what? Endocytosis, in case of if you're asked. All right. So look at this question. Which of these is an example of active transport? Which of them is an active transport? Come on, guys. Let's go. Let's go. Let's go. The diffusion of oxygen from a place of higher concentration to a place of lower concentration. The facilitated diffusion of potassium. The transport of glucose down its concentration gradient. The transport of sodium ion from a place of low concentration to a place of higher concentration. The transport of chlorine following its concentration gradients. Which is the answer? Ah! Somebody said pina colada. Pina colada. The day I drank that, it was turning my eyes. Hey, God, pina colada. Hey, God, you're very funny, man. Very funny. Pina colada. Quickly, guys. Quickly, 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 quickly. What was the answer? Somebody is still thinking. Okay, so that answer is D. All right. D. D is the transport of sodium. When sodium is moving from a place of low concentration to a place of higher concentration, like when it's moving from the urine into the blood, that is in terms of absorption. In terms of reabsorption, that is an example of what? An active transport. Why is it an active transport? Because it uses what? Energy to move. Remember, when sodium is in that vasculature, all right, in between the urine and the blood, and it wants to enter inside the blood, what does it do? It drags potassium inside and jumps into the blood. And initially, sodium entered there from the urine, like the filtered substance, with using what? Glucose. That's sodium glucose transporter. That one is not an active transport, all right? Because they agreed to marry each other. Then I entered the vasculature. Then, because sodium is a very wicked person, glucose what? Threw away sodium and jumped into the blood. Now sodium is confused. It needs to enter the blood. What does it do? It drags potassium inside and jumps in. All right. So that dragging of potassium, right, to jump in is known as what? Active transport. Okay. But that sodium glucose movement is not active transport. That's a normal transporter. You understand? It's not an active transport because it didn't use energy. It did not cheat. So I have two more questions for you on cell and I move. Yes, you're correct. Two more questions to go for me. Then I'll do my second advert. Hey. Don't get angry. The clothes is really nice, honestly. If it's not, I will not advertise it. Shut, I've never done any advert for anybody. But I really love the stuff. And this thing has been given to me since almost 3 to 4 months. Okay. Both active transport of a okay, no. What do facilitated diffusion and active transport have in common? What do they have in common? What do they have in common? Both involve transport of a solute up its concentration gradient. Both involve transport of a solute down its concentration gradient. Both involve the use of energy provided by ATP. Neither require ATP to function. Both are examples of carrier-mediated transport. What's the answer? Quickly, guys, quickly. Are we slowing down? What's happening? Are you hungry? Hmm? Come on, guys. What do facilitated diffusion and active transport have in common? Yes, you guys are so correct. The answer is E. Both of them are what? Examples of carrier-mediated what? Transport. All right? Last question. Last question. The sodium-potassium pump transports both sodium and potassium. Dash their concentration gradients in a process called what? Look at the option. In a process called what? Is it up? An active transport? Is it up? Facilitated transport? Is it up? Co-transport? Is it down? Active transport? Is it down? Counter transport? What's the answer? What's the answer, guys? Quickly. I'm seeing only 3 attempts. Where is everybody? Where is my... we are more than 30 here now. Come on. Energy. Oh, boy. You guys are wasting time. Oh. I'm not the one that is slowing down now. It's you guys that are slowing down. Jesse Transit. Is this person in the wrong room? He just transporting people to McDonald's. If you can get fries with high sodium and salt level on the fries, which creates high blood pressure. Please, what is the answer to my question? Okay, so guys, that answer cannot be co-transport. Why are we saying co-transport? Co-transport means that they are moving together. Do you understand? That's what co-transport is. This is just up and active transport. Don't you remember that sodium and potassium, they move opposite each other? You understand? It's an active transport. I describe that is exactly what I described in the last question. You see, that last question and this question is exactly the same thing. It's just that I don't know if it's a little confusing. All right. So please, the answer is up and active transport. Up, they move up their concentration gradients, which everybody agrees with, and it is what an active what transport sodium and potassium. The same question with last time. All right. I hope somebody say, okay. Okay. Exactly. Thank you. I know that's what you are trying to describe. Okay, you know what? I'll give you guys a bonus question. I'll give you guys a bonus question because majority of you failed this particular question. Look at this question and tell me the answer. This is not what I wanted to post. I wanted to post the options. Probably didn't copy it. So what's the answer, guys? Is it tiny? Yes. Let me increase the fonts. What's the answer, guys? Sodium and glucose are transported together from the intestinal lumen into an intestinal cell. The carrier protein is a dash, and the process is called dash. Quickly, 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 quickly. Everybody. I need everybody to make an attempt. Quickly, guys. Yes, you guys are so correct. So symport and what? Co-transport. You see the difference? You see the difference between co-transport? They are moving together. Oh, you understand? Couple, we are in love. But the other one, they don't want to use eyes to see each other. They are moving against each other and they are using power. That's why it's an active transport. Okay, guys. So that will be that for cell. Let me take notes, please. Okay. So second advert. Instagram, bad plugs for this shirt. It's very cheap. Honestly, you will not believe it. Trust me. You can even tell her to customize it for you. She will write your name. You use it as your scrub. Instead of wearing all this scrub that is not so fine. Bad plugs. Okay, guys, so let's move. Let's move. I'll still advertise it again in the next 30 minutes. Somebody's saying, Nchebe, why are you wasting time? Not my fault. Perfect. So let's go to genetics quickly. You see how fast we move? Because you guys are responding fast. Genetics. I actually love genetics so much. Hold on. Okay, guys, so let's begin. Somebody's already setting question for himself. Oh. Say translation, transcription. Interesting. Okay, so let's go. IP maths. You guys have studied though. Hey, I am so happy to hear that too. Hey, this is interesting. Okay, so let's start. This is testing the microphone. All of the body's non-productive cells called dash usually have 23 pairs of chromosomes and are thus called dash cells. Quickly, guys, quickly, quickly, quickly. Quickly, 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 quickly. You guys are gingering and suddenly nobody's talking again. What happened? The gas has finished. What happened to the gas? Quickly. Yes, okay. Since you guys are not talking much, I take it that we are just entering genetics. That's why everybody's like shaking. Genetics, I'm going to give you a revision, 30 revision questions, all right? And you have to pay strict attention to genetics. Genetics is one of the major things you'll be meeting. All of the body's non-productive cells are called what? Somatic cells, all right? And they are diploid. That's 2N, all right? It's really important that you know this. What's the answer to this? Quickly, guys, quickly. Without option, what's the answer? This is the option: species DNA, population DNA, gene pool, genetic drift, phenotype. The alleles possessed by all members of a population collectively are called what? Quickly now, only 4 people are answering out of more than 30 people here. What's happening? 15 people has to answer for me to move. The faster the better. Faster the better. Even if you know it before, put an answer. If you don't know it, put 911. If not, I am not moving. Yes, the answer is gene pool. All right. Look at this question. Quickly. What's the answer? Mathematicians in the house. The allele for a normal hemoglobin H is the dominant to the allele for sickle cell hemoglobin H. A male and a female who are both heterozygous for sickle cell hemoglobin have a child. What is the chance that this child will have the sickle cell hemoglobin? Are we still solving? Quickly, guys. Quickly, guys. Good. The answer is 25. I just crossed it now. You know. So, you know, they mentioned that the dominant is H, capital H. For the sickle cell is small h, and these people are heterozygous. So if they are heterozygous, that means they are capital H, small h, capital H, small h. All right? Exactly. Thank you. Thank you. You just saved me from the stress of explaining it. Yeah. So it makes sense, right? Capital H, small h, capital H, small h. The two capital H crosses to give you double capital H. That's normal, right? Then the capital H and the other small h, it gives you H, capital H and small h. That's a carrier, but normal phenotypically. But genotypically, that person is a carrier. Then that small h of the first one and the capital H of the second one will give you a carrier as well. But the two small h give you what? HH, small h, which is what? A sickle cell patient. So that's what they're asking you. They're asking you the genotypic, you understand, arrangement of the entire thing. The phenotypic arrangement of the entire thing. Okay. So 25%, yeah. All right. So for people that didn't attempt that question, I hope you understand what we just did. So what is this called? Select questions from these genetics is just so difficult because there is just a whole lot of things that could easily come out. Look at this question. Eye color is caused by genes at multiple loci. The phenomenon is called what? What's the phenomenon called? Somebody is thinking. Don't look at your slide. I see you. I'm using my spiritual eyes to see that you're looking at your slide. Don't respond as if you are in the exam hall. All right. Eye color is caused by genes at multiple loci. The phenomenon is called dash. Pleiotropy, sex linkage, polygenic inheritance, incomplete dominance, multiple carriers. Quickly, guys. Answer as if you are in the exam hall. Quickly, guys. Quickly. Let's go. Quickly. Yes, the answer is polygenic inheritance. Not, polygenic inheritance is what? Is C. Yes. Yes. Polygenic inheritance. Look at this question, guys. Quickly, everybody should attempt this question. Hemophilia is caused by a sex-linked recessive allele. This means that, what does it mean? You have to attempt this thing. Don't do yourself a if you are not attempting it, you are doing yourself a disservice. All right? Because if you attempt it, I want you to assume that you are in the exam hall. This is the exam preparation. All right? This is recap. It's not time for you to be writing anything. You don't need to write anything. You need to be, they put the options. Pakam, this is my answer. Nothing. You understand? You be gingering, energy. Too much energy should be worrying you. Quickly, guys. Females can be carriers but cannot be get hemophilia. The gene is found on the Y chromosome. A male can give the allele to his male child but not to his female child. A male who has hemophilia inherited the allele from his mother. A male child who has hemophilia inherited the allele from his father. Yes, the answer is D. A male child who has a hemophilia definitely inherited it from the mother. Justice for the men. Justice for men. Guys, give me the number to this. I'm not giving you option. Quickly. Let me see people that are active, has been active, intellectually active. A cell finishing mitosis has dash DNA molecules. Why a cell finishing DNA replication has dash DNA molecules? Quickly, guys. What are the two numbers? Quickly, guys. Quickly, guys. Okay, that's fair. So the answer is 4692, all right? If you don't know how we got the answer, please go to my video on genetics and watch it. You have time to do that. You really have time to do that. Better you understand it very, very well, than not. What's the answer to this? These are the options. Which of these is not found in the karyotype of a normal human male? 46 chromosomes, 23 pairs of homologous chromosomes, X and Y chromosomes, 46 chromatids, 92 DNA molecules. Quickly, which one is not? Yes, 4692 is correct. Correct. So, but we are in another question. Which of these is not found in the karyotype of a normal human male? Quickly, guys. Quickly. Quickly, guys. Is it because they have XY? Yeah, male, they have XY now. Which of these is not found in the karyotype of a normal human male? Quickly, guys. Quickly. It's everybody. Okay, yes, the answer is 46. All right. So they say which of the which is not found in the karyotype of a normal human male. What you are seeing in the karyotype is not chromatids. What you are seeing is chromosomes. What you are seeing is chromosomes, not chromatids. All right. So that's what they are just trying. That's why that answer is wrong. All right. So it's not about the number, but it's about the chromatids. And this is karyotypes. Because somebody is asking me, is it because they have XY? They actually have 46 chromosomes. They have 23 pairs of homologous chromosomes. They have X and Y chromosomes, the same thing. They have 92 DNA molecules, the same thing. All right. So, but they don't have 46 chromatids in a karyotype. They are specific about it. Remember what karyotype is? Karyotype is that chart, all right, where you see every. Feel me? Okay. So now look at this question, which is exactly very similar to what we were talking about before. Look at this question. A karyotype shows the dash chromosome sorted and isolated from a cell in dash. You see? So karyotype shows chromosomes, not chromatids. Yeah. Look at the options. Let's go. Okay. Somebody's already ahead of the queue. Interesting. You guys are very prepared, though. Why are we even doing revision? I need everybody to attend to this question. If you are not able, write 911 so that we will know how to help you. Yes, the answer is D. All right. The answer to that question is D. Sorry, I can't wait anymore. The answer is D. It's 46 and metaphase. Everything is arrested in metaphase phase. All right. Look at this question. Look at this question. Yeah, 46 and metaphase. That's D. Which of the following omits one stage of the cell cycle? Which of the following omits one stage of the cell cycle? You guys are doing quite well. Yeah, I'm impressed. I'm impressed. For real. Like, quickly, guys, quickly. Yes, you guys are so correct. Which of the following omits some stages of cell cycle? G1, S, G2, mitosis is correct. All right. That's then we have interphase mitosis complete. We have G1 DNA duplication and G2 mitosis very complete. All right. Then we have interphase, prophase, metaphase, anaphase, telophase, cytokinesis, very complete. But this one says prophase, metaphase, anaphase, telophase, cytokinesis, but there's no interface. So that's incomplete. You guys are doing quite well. The answer is D. I agree with each and every one of you. Okay. Chromatin shortens, thickens, coiling into compact rods during which phase of mitosis? Quickly, guys. No option. Which phase? Which phase of mitosis? Yes, D for that answer is correct. Quickly, guys. Yes, the answer is prophase. All right. Prophase, remember, prophase preparation phase. Prophase preparation phase. Prophase preparation phase. After that, everybody goes to the middle. That's metaphase. All right. Everybody goes to the middle. Then after that, we start dividing. That's anaphase. Then we start going back to the extremes of the poles. That's what? Telophase. Then we divide the entire cell. That's cytokinesis. You cannot forget this. All right. This is for life. Anytime you remember, you say, Nchebe taught me well. Like the next question, I'm not going to post it. It says sister chromatids are pulled to opposite sides of the cell during what? They break into two, they move. That is what? Anaphase. Remember that, because they may ask you something like that. I just don't want to post it so that we don't waste time. Exactly. Exactly. Now look at this question. Somebody is very radio, like somebody is active and engaged. Look at this question. Which of these processes occur during a cell's first gap G1 phase? All right. DNA is semi-conservatively replicated. The nuclear envelope breaks down. The cell synthesizes the enzymes that control mitosis. The cell synthesizes proteins and grows. The chromatin condenses into visible chromosomes. Yes, you guys are so correct. That is the cell synthesis, that the cell synthesizes proteins and grows in that particular phase. So you need to remember it. Now, there's a question that is asking about the stage where chromosomes aggregate along the equator. I can't give you this in this review. It's wasting time, because all of us know that middle M for M, all right? That's metaphase. That's when the cells come to the equator. M for M. M for M. Look at this question. Yes, metaphase. Look at this question. Yes, everybody. Why did we reduce to 20 something? Quickly, guys. Dash opens up one segment of the DNA helix during replication, and dash adds complementary free. You are sure you are going to get a question on enzyme, all right? And dash adds complementary free nucleotides to expose the DNA strand. We have the DNA ligase, DNA helicase, DNA ligase and DNA helicase, ligase and polymerase, ligase and polymerase. Okay. Then helicase and ligase and DNA polymerase and DNA ligase. Quickly, guys. Quickly. Quickly, guys. Yes. Yes. Helicase opens up, unwinds, right? It unwinds. Why polymerase helps to photocopy. So we have DNA helicase and DNA polymerase. That's B. So if you choose B, jump up. Yes, look at this question. Let me see. This question I failed. I always never ask this question. But now I feel like you need to know this question. Look at this question, guys. DNA replication is called semi-conservative. Semi-conservative. Why? Interesting. Somebody's really very active. Somebody is really very ready. Too much readiness is worrying this person. Come on, guys. What's the answer? Quickly, guys. Yes. So semi-conservative, all right? Semi-conservative means that each daughter cell has a new helix and an old helix. So somebody said kept one strand is kept. Exactly. So they break it into two, like pew, right? That each daughter cell, right? They will add a new one to it. So it has one new, one old. That's just what they're talking about. If you see it in the exam, remember it. That's if you see it. All right. I think the time for genetics has passed. So I'm going to stop genetics soon. Right? That's passed, right? But there are some important. Look at this question, guys, quickly. What is a mutation? What's a mutation, guys? Quickly, quickly. This is a giveaway. Come on. This is a giveaway, guys. This is a giveaway. This is a giveaway. What is mutation? Yes, you guys are so correct. Mutation is when the DNA structure changes. All right. And sometimes mutation can be beneficial, harmful, or neutral. All right. Like me, my own, I don't know if it's harmful or beneficial. I have mutation. All right. Lactose intolerance is usually mutational, especially if it's congenital. It's usually mutational. It's a mutation of short term of chromosome 6. All right. So, but somebody is telling me that I'm missing out. I don't know what I'm missing out. Honestly, I don't know how it is. So, how am I missing milk? But there are some that are harmful. There are some that are very beneficial. Some in the lab, sometimes we try to knock in and knock out genes. All right. Mutate genes. Let's see what it will give. You understand? Most stuffs we do in the lab is actually mutation. All right. So, but let's not overstate it. So, cytokinesis overlaps with which phase of mitosis? Which phase? The S phase, telophase, metaphase, prophase, interphase. Which particular phase? Which phase, guys? Which quickly, quickly. Exactly. You guys are so correct. So it's telophase, right? Everybody is so correct. Look at this question. All of the following participates in DNA replication, except we have the DNA, we have ribosome, we have DNA ligase, we have the DNA helicase, we have the DNA polymerase. Which one does not participate in replication? Remember, we have replication, we have transcription, and we have translation. You need to know where each of them occurs, all right? You need to know where each and every one of them occurs. So how we cook that protein, how we get the code to go to the kitchen, bring the amino acids, that's the ingredients, and prepare and organize that protein in the kitchen. That's what this thing is about. Feel me? Right. So which of the following does not help in the DNA? Remember where the DNA is. The DNA is the lab. No, is it the lab? What am I saying? The library. I hope my pronunciation is right. The library. Quickly, guys. Yes, ribosome. Why? Because ribosome is the kitchen. Brooklyn. Ribosome is the kitchen. That's where we cook all those things. That's where we cook the protein. All right? If you don't remember anything, you cannot forget this. You can't. I can't be giving this one. This question is the giveaway. I'm about to stop genetics now. Next 7 minutes. The G1, the S, the G2 phases of cell cycle are collectively called what? Quickly. Yes, telophase is correct in that question. But without question telophase, then after that, we ask all of the following participate in DNA replication except, and the answer is what? Ribosome, because everything that is mentioned there happens in the nucleus, right? Which is, and you remember that in the nucleus, that's where we do what? DNA replication. Then we come out from the nucleus into the cytoplasm, then that's where we now do what? Translation. All right. So the G1S, G2 phases of the cell cycle are collectively called what? You guys are so correct. The answer is interphase. You guys are so right. So the DNA polymerase is most active during which phase of the cell cycle? We have the S, we have the G1, we have the G2, we have the M, we have the G0. When does the DNA polymerase, when is it active? The S, the G1, the G2, the M, the G0. Quickly, guys. Quickly. I'm expecting everybody to be putting answers recklessly. Keep going. DNA polymerase is most active during which phase of the cell cycle? Keep going. Yes, you are so correct. The answer is S phase, not G phase, all right? Not G phase. S phase, that's where the DNA replication happens. And what causes DNA replication? DNA polymerase. That's the sense in it, all right? Yes, it's S phase. How many minutes do I have for? 4 minutes. Be done with genetics. 4 minutes. Okay. Look at this. Oh, I thought I was still sharing. This is the options. What is the product of translation? A newly synthesized amino acid, a newly synthesized polypeptide, a newly synthesized nucleic acid, mRNA, an mRNA, a tRNA. Answer. Answer. Please, now you guys should be faster because yes, you guys are so correct. The answer is B. It's a newly synthesized what? Polypeptide. That's a protein. All right? A protein. Look at this question. Which of the following is the template for transcription? Remember transcription, guys. Sticky notes. Transcription. The molecule that carries codons, the molecule that carries anticodons, the molecule that carries base triplets, the molecule that carries amino acids, the molecule that carries tRNA. Quickly, guys. Quickly. I need you to be doing this as if you are in the exam hall, all right? Don't open your slides. Quickly, guys, quickly. Yes, everybody. They said C. I'm waiting for you guys, so you guys are wasting time. See, I have 4 minutes. I would have given you like 4 questions, but you are not talking. So, yes, the answer is C. The molecules that carries the base triplets. Look at this question. The gene coding for a polypeptide made of 51 amino acids would have a minimum of dash bases. How many bases, guys? How many bases? What's the minimum number of bases we have? Quickly, guys. The mathematicians in the house. Yes, you guys are so correct. 153. You guys are so correct. Three plates. That's 333. Just multiply it and you get your answer. You guys are so correct. 153. Look at this question, guys. You were able to trace actively tag. You were able to radioactively tag and thereby trace an amino acid that is used to make insulin, a hormone that will be exported out of the cell. The pathway of the tagged amino acid would be what? You were able to radioactively tag and thereby trace an amino acid that is used to make insulin, a hormone that will be exported out of the cell. The pathway of the tagged amino acid would be what? So somebody is active and gingered. Too much energy. The energy is hitting me up. Is it energy or this? You guys should remember to patronize my friend. Patronize my friend. I'm advertising for her. She gave me this clothes. I didn't know this clothes was so good, and it's very affordable. Remember, bad plugs. Bad plugs. Let me type it. BADPLUGZ. Patronize her. I'm wearing the clothes free of charge. I did not pay. That's the dividends of adverts. Okay. What was the answer? Yes, the answer is B. Because it's a protein. Where do we produce? Where is the proteins made? In the ribosome. Where is this ribosome found? In Brooklyn. That's rough endoplasmic what? Reticulum. Because Brooklyn is rough. So rough ER. Then after that, the protein is being made. We take it to the dining and arrange it. That's the Golgi complex. Arrange it, fold it properly, package it. Then after that, we push it out to the Golgi vesicles. The waiters, people that are carrying it, the Golgi vesicles. Then from the Golgi vesicles, we push it outside. Extracellular what? Exocytosis, extracellular fluid. That is how it moves. It's as simple as that. They're just asking you the pathway of protein synthesis. Feel me? Okay, so I'll be asking you the last. I'll be asking the last question. Yes, the last question on genetics, because this is 12. Last question on genetics. I hope you guys are not feeling sleepy. But why will you feel sleepy when I am still awake? No, I should be the first person to feel sleepy. The genetic code is the link between the dash and the dash that they represent. What's the answer? This is the last for genetics. What's the answer? Genetic code is the link between the dash and the dash that they represent. Quickly, guys. Quickly. Oh, please. I'm sorry, but I still need to give you this one question. Let it be the last. I'm sorry. Thank God. You guys should talk now. It's just 4 people here, and we're more than 30 people. Come on. If you don't know it, just write not sure. I need people to type. I need you to really literally type. Because if you don't type, you sleep off. Everybody. I'm waiting, guys. Yes, the answer is E. I need you to actually type, all right? It encourages me to talk. mRNA, 20 amino acids, that's what it codes for. You have to remember that. You have to remember that. So this is the last question, guys. This is the last question. It was supposed to be the other one, but I just saw that I didn't, I forgot this. Quickly, guys. Since there are no genes coding for carbohydrates, nucleic acids, or lipids, how do cells produce them? How? Enzymes encoded by genes synthesizes these products. Cells are born with all the products they need. Cells absorb these products by endocytosis. Cells export these products from older cells. Cells can produce carbohydrate, nucleic acids, and lipids out of what? Proteins. Interesting. Yes, you guys are so correct. You guys are so correct. The answer is enzymes encoded by genes synthesizes what? These products. So we are going to the last chapter for today. Yes, 5 chapters is okay. We are going to, is it 5 or 4? But, it's okay. We will do 5. This one as the last one today. Then we'll finish up on Sunday. But bear in mind that I'm not going to do the last 2 chapters. I've already done videos for them. Posted the first one yesterday. Posted another one today. Please don't play with it. Remember, it's the latest topic and you've not been tested on it. Meaning that it will represent more questions than every other chapter because you've never been tested on it. All right. So those videos, watch it 5 times. As far as you are in this revision, that's good. But watch those videos 5 times. Thank you so much. Okay. So watch it very, very much. All right. You know, you guys are nurses. So I want everybody to pass. I want everybody to do well in your exams. So that when you buy your new car, you come and pick me, or you buy one for me. Okay, guys, let's talk about tissues. All right, remember tissues. Tissues is extremely important. It's one of the most important topics you are going to do because everything about you is tissue. Make sense? Everything about you is tissue. So let me test the microphone. Let me know if you still remember. All right, not like this question is a big deal, but it's just a test of the microphone. Let's test the microphone, guys. Testing the microphone. Testing the microphone. Pay attention. I'm not posting the answers. This is very cheap. The 4 primary tissue types found in adult organs include all of the following except dash tissue: connective tissue, fibrous, nervous, epithelial, and muscular. So if you know the main types of the primary types of tissue, you'll be able to know which one is not there. You feel me, right? You know which one is not there. So we have connective, we have fibrous, we have nervous, we have epithelial, we have muscular. Is it nervous or nervous? My pronunciation is terrible. Pardon me. Come on, energy. I need energy, guys. 10 people have to attempt this. Oh, I didn't. The other one was genetics, right? Quickly. What was the answer? Yes, fibrous. So the 4 primary tissues are the connective tissue, the nervous tissue, the epithelial tissue, and the muscular tissue. And you need to know the division of these tissues. Please, I will beg you again. I have tissues video. Go and look at it so that you understand tissue very, very well. You understand? If you're asking a question and you understand it very well, you'll be doing exceptionally well. All right? Now it's not time to read textbook or slides. Now it's time to attempt questions. Questions and questions. All right? It's being strategic. Okay, so I have 25 questions for you here. Now, I'm not asking this question. This one is talking about clear gel inside the cell. That's cytosol, all right, or intracellular fluid. So look at this question, guys, quickly. So this question is in different phases, all right? Okay. The dash gives rise to the dash. We have the mesoderm, digestive glands, endoderm, muscular system and skeletal system, ectoderm, nervous system and epidermis, endoderm, mesoderm, endoderm. Answer. Quickly, guys. Quickly. Come on, give me that energy. Give me that energy. Do you still remember about your mesoderm, ectoderm, endoderm? Do you still remember? Energy, guys. Energy. Energy, guys. Yes, you guys are so correct. The ectoderm gives rise to the nervous tissue and the epidermis. Makes sense. It's very clear. When you're talking about endoderm, inside, you're talking about the digestive system. Like that first option says mesoderm. Mesoderm is not digestive system. Digestive system is endoderm. All right. But when you're talking about mesoderm, that's the middle layer, right? You're talking about bones, you're talking about blood. Mesoderm, bone, blood. Endoderm, that's the inside one. You're talking about digestive organs. All right, digestive stuffs. But when you're talking act outside, you're talking about nervous tissue and what? Epidermis. You cannot fail this kind of question. How about this? Oh. Look at this question, guys. What's the answer? What's the answer? The middle primary germ layer is called the dash, which gives rise to the gelatinous material called dash, which then give rise to the different types of connective tissue, such as the bone, cartilage, and blood. Quickly, guys, quickly. I even explained this thing in the last question. Come on now, why are we wasting time? This is the last for this night, all right? You guys are not giving me the energy that can make me go more, honestly. So if I finish tissue, that will be it. You understand? And tomorrow I'm expecting the full house. So you have to talk to everybody. Because we also do pictures tomorrow. And tomorrow will be longer. But if you do very well tomorrow, we can finish up. If not, if you give me low energy, we end it. All right? So please, I really need that full energy. I work with energy. And you know me now. So the middle layer, the germ layer is what? Is the mesoderm, right? Yes. And it gives rise to mesenchyme. Mesenchyme is one that now forms the bone, blood, cartilage. Yes, you guys are so correct. The answer is B. Now look at this question, guys. Very fast, eh? Look at this question, guys. What's the answer to this question? The replacement of damaged tissue with scar tissue is called dash. When a tissue is damaged and is replaced by a scar tissue, what is it called? Yes, B is correct for that particular answer. Come on, guys, let's go. Let's go there. Only one person. What's happening? Is this question difficult? This is a very straightforward question now. The replacement of damaged tissue with scar tissue is called what? The answer is fibrosis. The answer is not necrosis, neither is it apoptosis, neither is it gangrene. All right? Neither is it regeneration. It is what? Fibrosis. You need to go and look it up. Go and look at my tissue slide for you to be able to understand this. Now look at this. With regard to the healing of a wound, a skin wound, macrophages. What's the function of macrophages? That's also they're asking you. There are different things that happens in wound healing, but they are very specific about macrophages. Why? Because we say that macrophages, I say name, I called it. For people that have listened to my lecture, people that have seen my video, I called it a name. I called it what? The genitors. Huh? Right? You remember? So if you can remember why I called it the genitors, then you'll be able to get this answer. You will smile and you pick your answer. Are you smiling? With regard to the healing of a skin wound, macrophages dash. Quickly, guys, quickly. Yes, you guys are so correct. The answer is C. Macrophages phagocytize and digest what? Debris. That's why we call them what? Genitors. All right? Look at this, guys. The pseudostratified columnar epithelium of the bronchi in smokers may transform into stratified squamous epithelium. This is an example of DASH. You see, this is pathology. If you can recast your mind back to what I discussed, you will see that I told you that we have this particular thing happening pathologically and happening physiologically. In physiologically, I told you, children below puberty stage, their vagina is what? Cuboidal epithelium. But when they get after puberty, it has to get stronger, right? It has to prepare itself. And the vagina changes from cuboidal epithelium to what? To stratified squamous epithelium, right? That thing is called the same thing with this. All right, it's exactly the same thing, but that one is physiological. This one is what? Pathological. That one is physiological. This one is pathological. If you don't understand what I just said, go to my reproduction video. You will see it. You understand it very well. Don't worry, I have video for everything. Come on now, just 2 people since more than a minute I posted this. Or you want option? Okay, let me give you option. There are some things I should not be giving option, like, you should just generate it quickly. You can't be getting an answer from option. Oh, why is this thing misbehaving? Only 2 people answered. Everybody's waiting for me to post option. Okay. This is the option. We have neoplasia, we have metaplasia, we have atrophy, we have differentiation, we have hyperplasia. Oh yeah, now you don't have any option. No. You don't have any reason not to attempt to answer. I know you were quiet. Oh, God. Yes, the answer is metaplasia. All right. I told you neoplasia is anything cancer. I told you atrophy. You understand? Atrophy, is shrinking, like reduction in cell size. Then I told you about differentiation. That's specialization. Then we have hyperplasia. All right. But this is particular what? Metaplasia. All right. Okay. Hmm. Don't tell me you wait for option for this particular question I'm about to post. Don't tell me you wait for option. After 6 months of lifting weights at the gym, you notice that some of your muscles have increased in size. This increase in size is due to dash of muscles. Quickly. Due to what? Hyperplasia, neoplasia, hypertrophy, metaplasia, atrophy. I hope you know the difference between hyperplasia and hypertrophy. I gave an example. like when maybe I have someone here and we copulate and we have a lot of children, we increase the size in size by a number. Or maybe I decide to start eating, and I get so fat that I feel this room. You need to know which one is hypertrophy and which one is what? Hyperplasia. I'm not teaching. So the answer to that question is what? Hypertrophy. The answer to that question is what? Hypertrophy. You need to, because this have clinical relevance. So you must know the clinical relevance. All right? Okay. What's this? Quickly, guys. I have a few minutes to go. The membrane that lines passageways that open to the exterior environment is called the dash. It's called the dash. We have, okay, I didn't give you options. I know. I don't give option, man. Yeah, so thank you for the love. So this is the options. The membrane that lines the passageways that open to the exterior environment is called. We have the lamina propria, we have the endothelium, we have a synovial membrane, we have a serous membrane, that's the serosa, and we have a mucous membrane, that's the mucosa. Quickly, guys, quickly. Quickly, guys. Some people are wondering, this question is too hard. These are the things you've seen. These are the things you've discussed. That's the idea of the revision. The membrane that lines passageways that open to the exterior environment is called the dash. Come on, I'm not getting the energy I need. Yes, you guys are so correct. It's the mucous membrane, all right? You can never forget this. The mucosa, the mucous membrane. It lines everything, all right? It lines all the passageways, the cavities, that's mucous membrane. You can't forget it, all right? Look at this question, guys. Look at this question. The membrane that lines the luminal surfaces of the digestive, respiratory, urinary, and reproductive tract consists of dash. Simple squamous epithelium and dense irregular connective tissue, areolar and dense irregular connective tissue, epithelium and lamina propria, areolar tissue and lamina propria, simple squamous epithelium and areolar tissue. What's the answer, guys? Come on. Let's go there. If you are feeling sleepy, put your leg inside cold water. For real, we did that. Those days in medical school, we do that. Anything to be awake. Awake, yeah. Did I say alive? Anything to be awake. Message retracted. The membrane that lines the luminal surfaces of the digestive, respiratory, urinary, and epithelium and the reproductive tract consists of dash. Come on now, you guys are slowing me down. Only 3 people. You guys have to be up to 10 or 10 to 15 responses before I continue. If not, I'll keep waiting. Trust me. Because, yeah, if you're not responding, that means You're tired and we can easily stop. Okay, yes, the answer is C. It's an epithelium. It's very straightforward. The membrane epithelium covers everything, both the respiratory, the urinary, the reproductive, even your skin. It's epithelium. All right. And below the epithelium, remember we say epithelium is avascular. There's no blood, blood flow. So below it is the connective tissue. All right. That supplies it, which is what the lamina propria. Look at this question, guys. Quickly, guys, quickly. Yes, the answer to that question is C. The Dash glands, such as tear glands, have vesicles that release their secretions via exocytosis. Then Dash glands, such as oil-producing glands, secrete a mixture of disintegrated cells and their products. Quickly. You have endocrine, exocrine, we have mucous serous, we have exocrine, apocrine, we have mucous cutaneous, we have ecrine, holocrine. Quickly, guys. 10 people has to attempt it for me to move. Quickly, 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 let's go. Yeah, yes. The answer is E, ecrine and holocrine. If you don't know the difference, go and watch that video on tissue. Look at this, guys. Let me give you the option. You need it. Look at this. Dash secretes dash into dash. We have the goblet cells, mucus, the blood. We have the endocrine glands. We have the hormones. We have dogs. We have the exocrine glands. We have hormones. We have dogs. We have endocrine glands. We have hormones. We have the blood. We have exocrine glands. We have mucus. We have the blood. Answer. Yes, is correct for that question. I need 10 people. We just have 6 people here responding. I need 10 people to respond to that question. You guys lack energy, the energy I need today. What happened? And you guys are so few, 27. You know, I always like a full house. Yes, the answer is D. Endocrine glands secrete hormones into the blood. All right. Look at this. Somebody will say, Nchebe, you love this question. That's anybody that has watched the videos. They'll be like, oh, Nchebe, you love this question. Because I talk a lot about it. So how would the genetic inheritance of a gene producing abnormal gap junctions affect muscle tissues? How? All types of muscle tissues would function normally. All types of muscle tissues will function abnormally. Cardiac muscle and some smooth muscle would function abnormally, while skeletal muscle will be unaffected. Skeletal muscle would function abnormally, Smooth muscle and cardiac muscle will be unaffected. Skeletal muscle and cardiac muscle will function abnormally. Smooth muscle will be unaffected. Answer. Quickly, guys, quickly. Only 4 people. Come on. I need up to 10 people responding to every question. So my ancestors said the answer is C. Yes, your ancestors are correct because your ancestors, they know that cardiac and smooth muscle, they have what? They have gap junctions, but skeletal muscle does not have gap junction. So because of that, if there's a problem with gap junction, it will only affect those people that has gap junction, which is what? Cardiac and what? Smooth muscle. So the answer is C. All right? Yeah. The answer is C. If you choose D, it's fine. All right. But note that the answer is C, and this is why it's C. You understand? Cardiac and smooth muscle, they have gap junctions. Skeletal does not. So if there's a problem with gap junction, it will only affect people that has it, which are the cardiac and the smooth muscle. All right. Yes, the answer is C. So look at this question, guys. I'll read out the options. Which type of muscle tissue is found in the walls of hollow organs? We have the smooth, we have the cardiac, we have the skeletal, we have the organ muscle, we have the elastic muscle. Which type of muscle tissue is found in the walls of hollow organs? We have the smooth, we have the cardiac. I'm not pronouncing so well, though. I say smooth. Normally, I would say smooth. And people will be wondering, what am I talking about? Okay, smooth muscle, cardiac muscle, skeletal muscle, organ muscle, elastic muscle. Yes, the answer is smooth. Nchebe is now pronouncing things well. Interesting. My water, I already finished it, but I know that this discourse will not be that long. That's why I didn't bring water. We are ending very soon. So look at that question, guys. Astronauts in zero gravity are able to move food through their digestive tracts because, what's the answer, guys? Yes, the answer is A. Smooth muscle produces waves of contraction that propel materials through the digestive tract. You guys are so correct. Smart people. Very smart people. So look at this. Which of the following is not a category of connective tissue? We have the supporting connective tissues, we have the fluid connective tissue, we have the aerobic connective tissue, we have the connective tissue proper, we have the cartilage. Quickly. Yes, the answer is aerobic connective tissue, right? It's not part of the thing. So I like the fact. So you need to know the normal for you to know the abnormal when they give you that kind of question, all right? So look at this particular question. Which type of connective tissue is abundant in the walls of arteries and in the airway? So it's a giveaway. Quickly, oh. Yes, everybody. Is this sleep? Are you sleepy? You want me to stop here? Exactly. That's what I want to do. After this chapter, I leave you. I'm very fast today. Like, for God's sake, we did almost 4, I think, is it 4 or 5 chapters today? Which type of connective tissue is abundant in? But it depends on you guys. You guys are not answering very fast. If you are very fast, we would have finished. We would have. Yes, it's elastic tissue, all right? Extremely important that you know that. It's elastic tissue. Look at this one. What's the answer to this? An example of a highly vascular tissue is dash tissue. An example of a tissue that is not highly vascular is dash. Quickly, guys. I'll ask you 10 more questions and that will be it. What's happening now, guys? Now you guys are not making me feel sleepy. Yes, the answer is areola and cartilage, all right? If we're going to do this thing on Sunday, we have to have an agreement too, because this energy you're giving me, it's not the energy of people that want me to. You know me. You people should know me. I'm too much energy. Glycosaminoglycans, that's GAGs, help give the ground substance some of its qualities, including dash. Yes, that D is correct, areola and cartilage. But this one, glycosaminoglycans help give the ground substance some of its qualities, including dash. What was the answer, guys? Helping to transfer charges in the nervous tissue system, holding water and maintaining electrolyte balance, giving bone its brittle inorganic structure, providing the major structural support of tendons and ligaments, acting as an energy reserve for muscle contraction. Yes, you guys are correct. Holding water and maintaining electrolyte balance. Which of the following does not account for the gelatinous consistency of connective tissue ground substance? Which one? Glycosaminoglycans, proteoglycans, congruentine sulfates, hyaluronic acid, and collagen. Which one? 8 to go. 8 questions to go. Eight to go. Yes, the answer to that question is E, collagen. Very correct. Look at this question, guys. Oh. Okay, look at this question. What's the answer, guys? A connective tissue fiber refers to dash, a nerve fiber refers to dash, and a muscle fiber refers to dash. Quickly. Interesting question. Entire cell, organelle, tissue, organelle, entire cell, part of a cell, organelle, entire cell, complex of molecules, complex of macromolecules, entire cell, part of a cell, complex of macromolecules, part of a cell, entire cell. Quickly, guys, quickly. Energy, quickly. Quickly, guys. Yes, the answer is E, complex of macromolecules, part of a cell, entire cell. Okay. No, this is connected to you. Which component? Okay, so yes, that answer is E, complex macromolecules, part of a cell, an entire cell. Look at this. Dash is associated with rapid transport of substances through a membrane, whereas dash is associated with resistance to abrasion. I have 5 more questions to go, so let's hurry up. Quickly, guys, quickly. Don't wait for options. Attempt it without option. Dash is associated with rapid transport of substances through a membrane. Which one? Do you remember the particular epithelium I told you that is in the alveolus? What happens in the alveolus? Transport, right? Exchange of rapid transport of substances, like exchange of air, right? Good. So, whereas dash is associated with resistance to abrasion. Which one do you find in every hard place? Every hard place. Exactly. You guys are so correct. So simple squamous epithelium is with rapid transport, but stratified is always everywhere that is hard, like the skin, like the every the mouth, the esophagus, the anus, everything. Five questions to go. What's the answer to this? Are they waiting for option? Stop waiting for option. What's the answer? I'm not giving you option no. I'm actually trying to get the next question safe. That's what I will do. Yes, the answer is simple columnar. All right. Yes, the answer is simple columnar. You're very correct. Look at this question. Found in the urinary bladder, dash resembles dash, but the apical cells are rounded and not flattened. Quickly. Quickly, guys. Yes, the answer is A, urothelium. That's transitional epithelium and stratified squamous epithelium, all right? The urinary bladder, ureter, they have urothelium or transitional epithelium. Then you have stratified squamous epithelium, all right, which is found in a whole lot of surfaces. Very important that you know that. Five to go, right? Look at this. A brush border of the microvilli is found in the dash epithelium, which can be found in the dash. So they're asking you where you find the microvilli, what your epithelium is that microvilli on, and the entire thing, where is it on? You know, that kind of question. Oh, you found Nchebe in this apartment, which is in this street. That's just what they're asking you. Quickly, guys, quickly. Okay, I didn't give you options. No wonder. Yeah, answering. Oh, hold on, let me give you option, all right? This question needs an option. Yes, this is the option. The brush border microvilli. You should even know where brush border enzymes are. You know what the brush border enzymes are? Those little enzymes that breaks things to the least molecule, right? Like the food has, for example, carbohydrate has broken down to the disaccharides, like maltose. Huh? Aha. When maltose come there, it cannot just enter as maltose. It has to be broken down by the brush border enzymes. So maltose, for example, will be broken down into 2 molecules of glucose. All right. Lactose comes down. The brush border enzymes will break down that lactose towards glucose and galactose. All right. Some people don't have some of those brush border enzymes, so they will not be able to break down those things to the smallest limit, which is absorbable into the blood. So that's the idea of brush border enzymes. So where is absorption found? Yes. Let me see what you guys are picking. Yes, the answer is C. You guys are correct. C, simple columnar, small intestine. Simple columnar, small intestine. Very important that you know this. Look at this. Dash epithelium is the most widespread epithelium in the body, whereas dash epithelium is rare. Dash epithelium is the most widespread epithelium in the body, whereas dash epithelium is rare. Which one is the most widespread epithelium in the body? Quickly, guys. So I have 3 questions to go. I know some people are already missing me. That's fine. Somebody said right now, who is missing you? Good. I will say B. What's B? Stratified squamous, stratified columnar. I will say B. So you are not sure. Don't tell me in exam you'll be doing I will say. I will say. Anyway, you're very correct. I will say stratified squamous is correct, right? And I will say stratified columnar is correct because you can hardly hear stratified columnar. Do you understand? Because it's too rare. Yeah, I understand. So look at this. Look at this, guys. How many do I have to go now? Is it 3 or 2? Okay. Dash epithelium provides a moist and slippery surface and is well suited to resist stress, as seen in the dash. Quickly. Yes, the answer is E, non-keratinized and vagina. Yes. No, not B, it's E, all right? Oh, I think, are you answering the other question? Stratified squamous and stratified columnar. If that's, you're answering that other question, you're correct. But this particular question is what? Non-keratinized vagina. Yeah. E. Yeah, exactly. Okay, so is it final question or final 2 questions? Hmm. Okay. So final question. Let me give you the simplest question. Yes, you're correct. You're correct. He said the one with the vagina. Okay, look at this question, guys. Which of the following is not a feature of the epithelium? And which one is not a feature? Let me give you options. Last question. Last question. Ha. I don't understand. Last question, and you people are quiet. What is happening? Which of the following is not a feature of epithelia? It's not. So there are 4 there are features, and 1 is not. Interesting. Please remember to go on this thing and patronize my friend on Instagram. Yeah, back plugs. Every 1 minute, one person answer. Every 1 minute, one person answer. This is the final one now. Come on. Is it the final or the second to the final? Oh, let me see if I'm missing something. Yeah, I think D. What is thinking? Okay, you guys are so correct. The answer is D. All right, D is not a feature of the epithelium. Abandoned extracellular matrix, that's in connective tissue, not in the epithelium. All right, epithelium is polar. That's why it does not allow water to just pass through your skin, because it's polar. You get polarity. You know what polarity is? If you don't know what polarity is, go and where did I even treat that? Discuss it in chemistry. Yes, I have the video. I have a live I did. Don't the cells take up space? And extracellular, abundant extracellular matrix is that they are scattered. The cells are scattered, but epithelium is jam-packed. It's joined together. So that abundant extracellular matrix is for connective tissue. That's what I said. You understand? So, and they are talking about which one is not. Polarity is polar. It is avascular. It has a basement membrane, and it has specialized what intercellular junctions. All right. So the one that is not is abundant extracellular matrix. So thank you guys very much. We've spent almost 3 hours, and you guys are doing well. So we will continue. I need you tomorrow. Use tomorrow. Watch the 2 videos. Those 2 last videos. All right. I'm posting the last one now. Immediately after I'm done with you guys, I'll post it. All right. Thank you so much. One person that is always speaking for me. I appreciate. You've tried. Oh, thank you. Thank you. I'm blushing. You're welcome. You're welcome. So you guys are doing well. You see, it's the same thing that we've done before. It's just a recap. That's why I said when you are doing review with me, you don't need to hold your pen. You don't need to write anything. You don't need to look at. Just be answering the question as if you're in the exam hall. Use it to test yourself. For people that are preparing for board, you need these exams as well. All right, your board exams. You need this test as well. You know, and also I will encourage you to go and get question bank. All right. So try to click this thing. You see that in there? That symbol there. Click it. Please, if you are not subscribed, try to subscribe. All right. So that if I'm doing live next class, even if I'm not teaching you, you'll be able to see it. Do you understand? Click your notification bell because it will help you no matter who is lecturing you, no matter your country, where you are right now, no matter the school, no matter whatever. This is anatomy, physiology is physiology. All right. At the end of the day, anatomy is location, 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 structure, 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 and physiology is what function, function, function. Done. Now it's not time for textbooks or slide. Now it's time for go watch all those videos, all of them. Prepare very well before the next listen. All right. So see you guys later. I need to rest. I was very stressed today. Just came back very late this night. So I'll leave you guys. Thank you so much. Have a nice night. Bye-bye. Ciao. Yeah, good night. Thank you so much. I appreciate it. Try to follow the subscribe if you have not, and put the notification bell on. And when you watch the video again, please, I want you to write ready. All right? I gave an instruction at the end of those 2 videos. That instruction, I will know people that really watched it to the end. You will meet it at the ending. Tell your classmates that I'm not here, right? Good night. Yes. I'm going to stop the stream now. I'm going to end the stream. Good night, guys. Take care.